ladies and gentlemen, good afternoon. Thank you very much for sparing the time to come and listen to us. Andrew Hopkins is always a tremendous act to follow. What I'd like to do in the next 25 minutes or so is share with you why I've given the title for this talk, "We Urgently Need to Transform Target Discovery." And I would underline urgently, and I would underline transform. What I'll do is, first of all, I'll kick off and share with you my thoughts on why society desperately needs new medicines. I'll then share with you some challenges that I think we currently have in drug discovery. I'll share with you some thoughts as to why I think we've got these challenges. I'll share with you what I think is a solution. I'll give you a couple of specific examples that some of my colleagues have already talked about at this conference. And then finally, I'll share with you some of the other things that we're currently doing to try and change target discovery the way we currently do it. So first of all, let's think of society. I said society desperately needs novel medicines. I'm not talking about Me Too's and new formulations. We need pioneer medicines. Now, I'll give you some figures in the UK. In 2050, 27% of people in the UK will be over the age of 65. And I'm sure the figure is similar to the US. That's 1 in 4 people. It's very hard for many of us to imagine walking down the streets and 1 in 4 people are over the age of 65. And in Japan, that figure is actually close to 1 in 3. And of course, over the age of 65, the incidence of dementia goes up almost exponentially. The incidence of cardiovascular disease goes up almost exponentially. And the incidence of cancer goes up almost exponentially. We desperately need novel treatments for these conditions. The other challenge we've got in society is that in some parts of the world, we've almost got epidemics in certain diseases, what I would call maybe diseases of modern living. Diabetes, obesity. We desperately need new treatments. Another major challenge that we all face is when we currently have discovered truly novel medicines, they're too expensive. Let me share with you the example of Vertex. 3 years ago, Vertex launched a new drug for the treatment of cystic fibrosis. This drug works in 4% of patients. It works beautifully in 4% of patients. That drug costs $300,000 a year. That's $800 a day. Which individual and which payers can afford that? Frankly, in the UK, if we came up with a new drug for depression or schizophrenia or pain or Alzheimer's and we charged $800 a day, the NHS would be bankrupt overnight. These are major problems. So what are the challenges we have in drug discovery? Well, in drug discovery, I think the way we're currently doing drug discovery, I think it's unsustainable. Now, I'm sure many of you have seen the analysis that was published at the beginning of 2013, done by Forbes, where they looked at a number of large pharmaceutical companies. They looked at how much money they'd spent on R&D over a finite period. They divided that by the number of drugs that each of those companies had launched, and they came up with an average cost of a drug. That's not a bad way of doing it. Now, in that table, published by Forbes, January 2013, for AstraZeneca, the average cost for a new drug was $12 billion. Amgen were at the bottom of that table, and their average cost was $3.5 billion. Even $3.5 billion is unaffordable. It's unsustainable. And bear in mind that many of these drugs were not pioneer medicines. Many of them were Me Too's or new formulations. And what we need is pioneer medicines. Drug discovery, apart from being incredibly expensive, it's also incredibly risky. A paper came out earlier on this year where they looked at a number of drugs that were in development, so they were in clinical development, phase 1, 2, or 3, for cancer in 2003. In 2003, there were 529 compounds in development for cancer. And then they looked to see what happened to those compounds until 2013, May 2013, so a decade later. What they found was that out of those 529, 45 had made it to the market. That's great. 95 were still in development, but 389 were terminated. 389 were terminated, molecules that were in phase 1, 2, or 3 out of 529. This is the level of attrition that we have. Their analysis then went on to show that if a compound is in phase 1, what's the probability of it hitting the market? The answer was 7.5%, less than 1 in 10. If a compound is in phase 3, what's the chances of it reaching market? The answer was 33%. So even in phase 3 for cancer, only 1 in 3 molecules made it to the market. This is an incredibly risky business. You're all aware in the Alzheimer's space, and we're going to hear a talk on that in a few moments, but in the past decade, there have been 12 or 13 very large clinical studies in Alzheimer's. They've all failed. Many of those studies were phase 3 studies, and one of those studies actually cost $750 million, and it failed. So the process is expensive, it's risky, but it's also incredibly slow. A paper that my colleague Stefan Knapp published earlier on this year, he looked at a number of kinases which were associated with cancer, and he analyzed how long it took those targets or molecules for those targets to reach the clinic, so to reach phase 1. Altogether, he looked at 28 kinases, and for those 28 kinases to get to phase 1, it took between 6 and 30 years. 30. This is just too slow. In the pain area, I can tell you, in the early 90s, 91, 92, I was working on a specific sodium channel, which I believed was restricted to the dorsal root ganglia, and a lot of companies are still working on that channel, but 25 years later, the molecule still hasn't reached the market. In the mid-90s, there was data that blocking NGF was a great target for treating chronic pain. 20 years later, we still do not have a drug. In the early part of this century, 2001-2, there was some data from Boehringer Ingelheim that a small peptide molecule which blocks CGRP antagonists was an effective treatment in patients for migraine. That was 2001-2. In 2014, we still don't have a drug on the market. We are too slow. The process is expensive, it's too risky, and it's too slow. What are the reasons? I'd break the reasons down into maybe 2 categories. One, I would say organizational, and the others would be science. So let's tackle organizational first of all. One of the challenges that we have in both academia and in industry, currently there is massive duplication of effort. There are so many companies that work on the same few targets in parallel, in secret. It's not surprising because everybody reads the same literature, they go to the same conferences, and they talk to the same opinion leaders. They start working on that target. 6, 7 years later, they come up with a proprietary molecule, and they test it in patients for the first time. And the failure rate for a novel target in patients in phase 2 is more than 9 in 10. More than 90% of novel targets fail in phase 2. Now, for any one company, that is a tragic waste of money. It's also a tragic waste of people's careers. But when you think there might be 20 companies doing the same target in parallel, if one fails, the other 19 will as well. That is a horrendous waste of money. It's a horrendous waste of people's careers. But ladies and gentlemen, it's a horrendous waste of patience. The way we're doing drug discovery today, we are exposing patients to molecules that other groups already know are destined for failure. Ethically and morally, that is wrong. I can share with you a personal example. In 1997, when I was working in Glaxo Wellcome, I was working on an ion channel called TRPV1, TRPV1. I believed if I could block that channel, I'd get an analgesic. In 1999-2000, Dave Julius and my friend John Davis published knockout papers in Nature and Science. And they basically suggested that if you knocked out that protein, it looked as if it abolished pain, or it reduced pain. In 2000, the whole industry jumped on that target as an analgesic target. In 2003, when I was now in GlaxoSmithKline, we came up with a potent selective antagonist. We took it into patients, and there was no analgesia, but it actually increased body temperature. So we stopped it. In 2006, there were still 60 companies working on that target, 60. In 2010, I was chairing a pain meeting in London, and there were still academics, still biotechs, and still pharma working on that target. And then, December 2010, AstraZeneca published their clinical data. They got no analgesia, and they got an increase in body temperature. Now imagine in those 13 years how much money we wasted on that target. We're not going to get an analgesic, but also just think of how many patients we needlessly exposed. I think the other challenges that we have organizationally, and this is not meant as a criticism of industry. Industry are private concerns, but I do believe that I do worry about the fact that there is too much chopping and changing and not enough stability in some of these organizations. And of course, when something is difficult, you need longer-term commitments. In terms of scientific challenges, I do believe that in many diseases, our understanding of the pathophysiology is abysmal. We don't really have a clue what causes depression or schizophrenia. This is why 9 times out of 10, when we take a molecule into phase 2, it fails. For many diseases, we don't have good biomarkers. You can't ask an Alzheimer's patient if their memory is better today than it was last month. And likewise, you can't ask a depressed patient if they're less depressed today than they were last week. We need better biomarkers. We don't even know how some existing drugs work. Paracetamol, acetaminophen, probably tens of millions of people are going to take that drug today. We do not know what the molecular target of paracetamol is. If we don't know how existing drugs work, how can we design better ones? These are the scientific challenges. I also believe, and many of you know, I am not a fan of animal models. I think I have seen so many targets, so many molecules work beautifully in animal models, and we take them into the clinic and they don't do a thing. We have to recognize that there are limitations to these models. We will never, ever have a truly predictive animal model of Alzheimer's or schizophrenia or depression or chronic pain and many other diseases. So those are the challenges. So what's the solution? I believe the solution is we have to pool our resources. That's what we're doing in the SGC. We are working closely with 10 large pharmaceutical companies. Those companies are GSK, Pfizer, Novartis, Lilly, AbbVie, Takeda, Boehringer Ingelheim, Janssen, Bayer, and Merck. And each of those companies has given us $8 million over a 4-year period. We have $80 million of private funding. The other thing I think we need to do is in the early phase of discovery, let's be less obsessive about IP and secrecy and competition. Let's make our data, knowledge, reagents freely available. That's what we do in the SGC. All of our data, all of our reagents, all of our knowledge, we make it immediately available as soon as we've got it. Doing that will reduce duplication. It will reduce other people's wasting their time and effort and resources. Now you can imagine by doing this, as a consequence, every academic wants to work with us. Every academic who comes into my office wants to collaborate with us because they know I've got no secrets. I'll share all of our know-how, all of our expertise, and all of our reagents. And frankly, that transparency creates a lot of trust, which is great for collaboration, it's great for science, it's great for drug discovery. So consequently, we are now collaborating with more than 250 academic labs all over the world. The third thing that I think we need to do is we need to work on innovative targets. So again, that's what we do in the SGC. We work on protein families or proteins that other groups are not working on, because we believe that's where the truly pioneer drugs are going to come from. So let me share with you 2 specific examples. The first, and I believe many of my colleagues, Panagis, Stuart Conway, have probably talked about all of this already, David Owen from Pfizer. So we started a program 5.5 years ago to generate novel inhibitors for novel epigenetic proteins. We decided to work on a family of proteins called bromodomains. There's 61 of these in humans. Now, at that time, many people said we wouldn't do it because this is essentially a protein-protein interaction. Well, we did do it. My colleague Panagis, who's in the audience, actually published that in Nature in December 2010. He and the team with Jay Bradner and GSK actually showed that that molecule works in a rare cancer called nut midline carcinoma. That was December 2010. What happened after that in the past 3.5, 4 years? Well, that molecule that we generated, we have given out to more than 400 labs all over the world. And these labs have taken that molecule and they've now shown that it works in a whole range of other cancers. It works in a model of sepsis. It works in a model of cardiac hypertrophy. It works in fibrosis. And there's even a suggestion it could be a new target for male contraception. Now you just think of those 5 indications: cancer, cardiac hypertrophy, fibrosis, sepsis, and male contraception. What the hell has that got in common? Nobody would have predicted it. This is an example of a novel target, novel molecule, and at least 5 broad areas of biology. We are not good at target discovery, and this is what's killing this industry at the moment. Now, since then, I said we've given that molecule to 400 labs. Panagis' paper has been cited more than 500 times. There's been more than 200 publications, many using that molecule and on that particular target. Many of our pharma partners have started proprietary programs. There have been new biotech set up, one of them here in Boston. And this has been the impact of this one freely available molecule on science, on drug discovery, and enterprise. Today, 3.5 years later, there are 6 companies with 6 different molecules in the clinic. This is unprecedented. A completely novel target 3.5 years ago, and now there are 6 clinical studies ongoing by 6 different companies. Let me share with you another example. So Udo Oppermann, I don't know if he's in the audience, he's giving a talk tomorrow. He's a phenomenal scientist based in Oxford. He's been working with GSK, and he generated an inhibitor for a Jumanji demethylase called JMJ-D3. He published it in Nature, I think, about 2.5 years ago. He took that molecule and he put it onto macrophages and showed that it reduced the production of TNF. So these macrophages are throwing out TNF, and it reduced the release of TNF. Now, of course, you all know anti-TNFs are selling $28 billion a year. And this potentially is a new mechanism for reducing TNF production. He put it onto osteoclasts and showed that it reduces bone resorption. And he put it onto human breast cancer cells, and he showed increases apoptosis. So again, a new target, new molecule, 3 lots of data: inflammation, bone, and cancer. I do not think there's anybody on the planet who can tell me where that target will end up. I think the only way we're going to find out is by taking a molecule into the clinic. Now let me just share with you some of the other things that we're doing and the direction we're going in the SGC. So the first thing is, you're all aware that over the past 15 years, there's been some very large-scale genetic studies. There's been at least 3,500 genetic hits, but more than 90% of those 3,500 genetic hits have never been screened chemically. So what we're going to do is we're going to generate proteins and assays and structures and chemical starting points for some of those genetic hits, because we believe a greater proportion of those genetic hits will translate into new therapeutics or new targets for drug discovery. The second thing is we're now building human platforms. So we're accessing primary human cells from healthy individuals and patients. We're focusing on inflammation. And as we now generate these novel inhibitors, we're going to systematically screen them in primary human cells. We believe this is going to be a much better way to generate new targets for chronic inflammatory diseases. This will allow us to compare healthy with disease, disease with disease, cell type with cell type, and target with target. The third thing we're doing is we're about to set up a group in Brazil to generate novel kinase inhibitors for novel kinases. And again, all of these reagents will be made freely available. We've also just got funding to set up a drug discovery institute in Oxford focused on dementia. There, what we're going to do is we're going to take novel dementia targets, we're going to generate novel inhibitors that we can use in cellular assays, but we're also going to generate novel inhibitors that you can use in vivo, potentially in animal models. And all of those will be made freely available. But because we believe target validation happens in patients, it doesn't happen in animals, and it doesn't happen in cell lines, what we're intending now to do is we're going to take some of these targets, we're going to generate clinical molecules, and we're going to take them all the way into patient studies, but completely in the open. We're going to identify that 1 in 10 target that has the potential to do something in patients. And we're going to identify the 9 in 10 that is complete garbage. So in summary, what I'd like to say is I believe that in early discovery, secrecy and competition is wasting money, it's wasting people's careers, and it's wasting patients. It's slowing down drug discovery at a time when we desperately need new medicines. Let's work together, let's identify those 1 in 10 targets, and then industry can take those 1 in 10 targets and generate proprietary molecules and take them all the way to the market. That will be good for industry, it will be good for patients, and it will be good for society. Ladies and gentlemen, thank you very much.